I want to thank AstraZeneca for inviting us here. We're very excited to present GREmLN, which is the first single-cell transcriptomics model for genomics that's transformer-based and uses gene regulatory networks as input. So quickly, I'll just introduce the speakers again. So we have Ming Zhang. Unfortunately, he can't make it today, but Ming is a PhD candidate at the Califano Lab and Azizi Lab at Columbia, and he is largely the AI lead for this project. And we're also joined by Vinny Swami, who's a PhD candidate at the Al-Karashi Lab and Ramadan Lab at Columbia. And then we have Rowan Cassius and myself, Leo DePierre, who are AI staff associates at the Califano Lab. And quickly, before we get started, I want to thank Damien Dikil for the invitation. I met him at ACR in Montreal for the AI and ML meeting, and so I'm happy our conversation led to this occasion. And I also want to thank you, Elizabeth, for helping organize this talk. So first of all, the program. So these are the topics we'll be going over today, starting with the motivation, then the data, the introduction to GREmLN, and then the methods, followed by benchmarking results and some conclusions and future work. And also, I was told at the beginning of this call that last week's talk was on the limitations of single-cell foundation models. So we're excited to hopefully, shed some light on the exciting parts of these foundation models, and also new ways to basically implement gene regulatory networks and gene regulation into them to improve their performance. So starting off with motivation, so the long-term mission is that of virtual cell models, or VCMs for short. And essentially, these would allow us to run experiments in silico with rapid simulations rather than doing tedious, time-consuming wet lab work. And so that also means that we can select hypotheses to test in the lab based on their likelihood of success, and that will ultimately save resources and time. And you can also use it for other tasks like fine-tuning to an individual specific cancer or disease context to find targeted therapies, among many other uses. And for instance, how cancer chemistry changes with treatment. The right treatment may no longer be effective after the wrong one is administered. And so with VCMs, the idea would be, for instance, that we could run different simulations ahead of time to identify the right treatment from the get-go. But again, like I said, this is, there's a whole variety of things we could do with this. And ultimately, the point of VCMs is to accelerate scientific discovery and have therapeutic implications down the road. Now, GREmLN is a piece of this puzzle with the Virtual Cell Project at the Chan Zuckerberg Initiative. So we're very excited to share this work. And what this means for the short term is that right now we're focusing on kind of stepping stone kind of tasks like perturbation prediction, drug sensitivity prediction, cell state control, or cell state transition control, and generating rich embeddings for other methods to use. And of course, along the way, we want to have as much explainability as possible, and so we'll talk about that in some of the future work later on, but it's not always attainable. But why do we use gene expression? There's proteomics, there's epigenomics, why are we sticking to gene expression? So gene regulatory logic controls cell states. And in cell biology, there are stable low energy endpoints defined by gene regulatory networks. I'll also refer to gene regulatory networks as GRN throughout the talk. And these GRNs are predictive of cell states. And phenotypic plasticity can be modeled as multiple stable low energy end states. And this plasticity can lead to the resistance to therapeutics. And also, this originates from multiple sources like genomics and also non-genomic sources. And to target therapy resistance in tumors and immune subpopulations, we need models that identify plasticity mechanisms that define cell states, identify important regulatory structures within cell states, and models that can predict molecular responses to genetic and chemical perturbations. Moving on to the data. our data consists of single cell RNA sequencing data. So as I'm sure you're all familiar with, so single cell gene expression data is almost like a cell sentence where each gene has an associated expression value. And essentially, we want to model the relationship between genes, much like a traditional LLM models relationships between words. And the pre-training data set that we're using is CellXGene. It's a very large corpus of single cell data, and it's curated by the Chan Zuckerberg Initiative. And we use only healthy human cells in pre-training. It's important to build this, solid understanding of how a healthy cell works in a more stable context before we start fine-tuning on erratic disease behavior that can be more noisy. And the pre-training data is on 11 million healthy human cells spanning 19,000 genes and 162 cell types across various cell types. Across various tissue contexts, excuse me. Now, one of the issues with this data is that genes on the surface have no apparent ordering. And that's noteworthy because transformer architectures benefit from this underlying structure of tokens. For instance, in language, in English language, for instance, the order of words in a sentence really matters. And it's, this is the stuff that transformers pick up on. And without a gene ordering scheme, we're effectively presenting a bag of genes approach to the model, much like in language, you would have a bag of words. And this essentially is like shuffling words randomly in a sentence and trying to find the meaning of that sentence with this jumbled up, these jumbled up words. And this can make it harder to find the meaning. And that's where positional encodings are often used in transformer tasks to inform the structure. So in language, it would be saying, okay, word A, or word 1 comes before word B, etc. So the task here is how do we inform this structure in gene biology? So previous work, or specifically transformer-based foundation models in the field, have different approaches. So SCGPT and SC Foundation both used unordered gene tokens. So SCGPT specifically randomizes the order of its genes so that the model never can pick up on this kind of sequential ordering. It's built to be robust against that kind of order and find relationships despite that. And SC Foundation just doesn't order its gene tokens either. And then finally we have GeneFormer, which ranks its tokens by their expression level, which also is quite arbitrary. It's almost if you imagine, ordering the words in a sentence based on how long they are, you won't really capture anything that's linguistically relevant. And so presumably that is the case as well in biology. And so we feel that there's this gain to be achieved by finding this relevant structure and adding this additional contextual information to the model or to the model's data. So again, what's missing? Well, as I said, the previous approaches underutilized transformers' abilities by limiting the structural context of gene tokens. And they do capture some gene-to-gene co-expression structure, and we'll show that in our benchmarking. They do, have some notable performance, but there's still a lot left to be gained, especially as we get to more complicated tasks on the road. And so right now the task is quite hard because the search space is immense. And so if we introduce regulatory graphs, we can provide more contextual, or we can contextualize the data and reduce the model search space and provide an underlying structure that benefits transformer attention. So quickly, I first want to go over what are these regulatory graphs. I'll cover GRNs, which is what we really focus on. That's what we use in pre-training. But we also have we've used PPI networks. So quickly, I'll go through these. So essentially, a regulatory graph captures regulatory interactions between genes or proteins. In the case of a GRN, so this provides a regulatory insight into a specific cell state. So the GRN is cell state specific, and it'll change with cell state. And on the other hand, PPIs are cell state agnostic, and they represent the molecular interactions and functions between proteins across all cell states, which kind of gives more of a general regulatory structure view, but not necessarily giving as much information as to what the specific cell state is. And we construct these using the Arachne algorithm developed by the Cloud Fauna Lab for the gene regulatory networks. And for PPIs, there are available networks out there like String and Hint, which are essentially the consolidation of different information, such as experimental evidence, computational predictions, and also literature-based semantic associations. Also, an important note when we're using these graphs is that these graphs tend to be very sparse and noisy. And so therefore, it's important to not constrict the model to these graphs. We want the model to attend to them, be able to learn from them, but also we're cognizant that the graphs don't capture everything, and so we also want to give the model some flexibility to learn around them as well. And so it can learn local gene dependencies from the graph, but also learn longer term gene dependencies that are not captured by the graph. And also this means that we can model potential, higher order group interactions instead of just pairs, which is what the graphs currently capture. Pairs of genes, that is. I'll quickly go through our pre-processing pipeline. So we run through different steps, such as general quality control. So that includes, UMI filtering, making sure that we don't have any doublets in our single cell data, and whatnot. Then we generate our gene regulatory networks using the Arachne algorithm. We also run highly variable gene selection to make sure that we're not including any housekeeping genes that aren't very indicative of cell state. And then we quantize our data into 100 expression bins, or we're quantizing specifically the expression. And that essentially allows us to tokenize it later on for use in our pre-training task. And finally, we also do selection of the GRN, but on a cell-specific level. So just subgraphing or subsetting the gene regulatory network of the cell type to the cells that are included in our cell. And we split our data set into two main categories, the first one being seen cell types. So this is what the model sees in training. So we have a training set that takes from the seen cell types, and then the first validation set is on these same seen cell types. And then we have our held out cell types, which are a validation set or another, almost like a test set, so we can test out of distribution performance of the model. And I just want to reiterate also that we never see cancer data. We're only testing on healthy human cells. And so that's interesting for later on in our benchmarking. Now, introducing GREmLN's architecture. So again, our motivation is that the other models in the space, we feel aren't taking advantage of this kind of gene structure or gene-to-gene interaction structure, which can add all this context to reduce the search space of the model. And the use of these large amounts of single cell data coupled with the regulatory networks that we've inferred can allow language models to learn reasoning that follows graph structures containing regulatory logic. And our modeling strategy is to use a large-scale inference of gene regulatory networks, and we encode graph structure into the attention mechanism of the transformer architecture. What that means is we're essentially altering our attention mechanism, and we'll get into this in later slides. But so we're not actually using a positional encoding like normal LLMs would. We're actually transforming our query, but we'll save that for later. And we're using mass language modeling as our pre-training task. So the model is also conditioned on graphs doing this mass language modeling. Moving up to methods. So we tokenize our scRNA data. And so essentially we split the incoming cell into two parts. So the gene ID and the expression. So we tokenize the gene ID, just giving it a certain basically identifier that the model then gets familiarized with, and the model can interpret what this gene is. And then we also tokenize the expression. So there's a separate representation of the expression level. And so we embed these, and so then we have a gene embedding and a rank embedding. Also, it's a rank embedding. It's also an expression embedding. We can use them kind of interchangeably. And remember that the expression is quantized as well. It's discretized into these expression bins. And there's no positional information yet, right? And then we simply concatenate these two into a larger embedding, a combined embedding of both the gene information and the rank information. And we feed this to our graph diffusion kernel attention transformer. And so our input embedding to the transformer is then twice the dimension, of course, of the individual gene embedding and rank embedding. And again, there's no positional information yet. And so this begs the question, how do we use graph to inform a positional encoding, and how do we implement it? So I'll hand it off to Vinny, who's going to be explaining that.
Hey, everyone. Okay, you want to go to the next slide, Theo? Thanks. Okay. Right, so Leah's talked a lot about how we want to essentially have some sort of positional information injected into our modeling process, and how we can't really use the conventional sequential-based positional encoding mechanism. So things like encoding the sequence length through some sort of sinusoidal activation, we can't really do that. And so what we end up doing is something, actually, could you go back one slide? Called heat diffusion, which is where if you can imagine, all graphs as being, all genes being nodes in this graph, we pass information between connected nodes in the graph through iterative steps, with the idea being that genes that are closely connected in the graph will have information aggregated between them, and genes that are not connected in the graph will essentially not be sharing information with each other. And so this essentially transforms the original embedding of the expression space based on the structure of the graph. Okay, now we can do the next slide. And the way that we do this is through essentially something called the kernel gram matrix. Essentially, we're computing eigenvalues of the graph Laplacian. The simple way of thinking about this is we're essentially smoothing out the graph and then finding out, what are the major sources of variation across the connected components in the graph. You can imagine a node that is connected to every single gene having very little meaningful information, because the node is going to be communicating the same information to every single other node in the graph, versus a gene that is, maybe has a few relatively small number of connections to other components in the graph, where that's information that we want to retain. And so we essentially take the GRN, apply this transformation, and then we multiply that to our query, so the actual matrix of gene expression. And the advantage of this is that it's very scalable. We are essentially performing a single transformation operation to the actual structure of the to the actual expression values. And the advantage of this is that it allows us to sort of have an arbitrary size graph that we can kind of subset. So, for example, when we're going from, let's say, one cell type to another, we're able to essentially manifest the entire graph, of all maybe 20,000 genes, without running into any sort of computational constraints. Next slide. And so as you can see here, in the traditional transformer attention, it's your query times key. Essentially, all we're doing is transforming the queue by first injecting this graph information via the heat diffusion, which is really just a series of matrix multiplication operations. And so the advantage of this is that we're still able to use the kind of standard attention mechanism that's well established to be very expressive, has great computational properties. And so this allows us to scale the model to many GPUs, large numbers of cells, and overall retain the efficiency that we kind of see in the traditional transformer modeling field. Next slide. And so essentially, we're able to kind of explicitly inject the graph information into the query here. And so one of the key takeaways here is that the graph must process the graph information. And so we are kind of having it as a sort of a strict dependency in that the model is always going to be seeing graph information. But because it's still doing general transformer interactions, you can still capture interactions between genes that are outside the graph itself. These are the things that it's going to have to learn. But the graph itself just creates sort of a soft bias that the model can sort of lean on while it's in sort of maybe the earlier phases of training before it can learn kind of these more complicated relationships. Next slide. And so functionally, this looks very similar to your traditional transformer network. You have your embeddings. We have which we project into a query key and value. We then inject our graph information via the graph diffusion matrix. And then from there, it's just a simple transformer attention layer. Our model right now is relatively small in terms of the transformer world. Just only 3 transformer blocks, 8 heads. And so we found that a relatively small model, as Rowan will show you, actually gives quite good performance. And so one of the main things to know here is that we can control, we have a bit of tunability on how much diffusion we apply to the graph, which we have tuned over various hyperparameter searchers to about 0.1. And so just to sort of reiterate this, the graph structure provides sort of a soft bias that encodes the gene regulatory network, again, which can be both a GRN inferred by Arachne, as well as PPI information, and then essentially encodes that into the query, which we then use for a standard transformer operation. Next slide. So this is just a quick little computational trick where essentially computing the eigenvalues for the heat diffusion is actually computationally very challenging. So we have this trick called the Chebyshev approximation, which allows us to do this in subquadratic time. Just a lot of math, but it ends up making our model a little bit faster with a small trade-off in numerical accuracy. Next slide. And so the way that we actually train this model is through a mass language modeling objective. So people are familiar with the BERT style model, where you take a sentence, you hide some of the words, and you ask the model to fill it in. We're doing something very similar, where we take our genes, we mask some of the genome embeddings and the expression values, and then we ask the model to fill those back in, conditioned on both the surrounding expression and then implicitly by the structure of the graph that we've added into the actual queries. And so by doing this self-supervised learning objective, the model is essentially able to learn, how do I fill in these pieces? What are the important surrounding bits of gene expression information that I need to know in order to fill these in? And you can imagine that for this task, a gene regulatory network or a PPI network provides a lot of very meaningful information. The model is able to see sort of softly what genes are influencing others.
Ground, if you allow me.
What was that? Okay, never mind. But yeah, the structure of the graph provides a very good sort of baseline for the model to be able to fill in these missing gene expression values. Next slide. And so yeah, this is essentially the same thing, but showing the math. But essentially, we're trying to maximize the probability of finding, of predicting the right gene expression token or the right gene embedding token, given the surrounding context, right? So the model is basically able to see, if you are looking at the diagram, the two gray pieces, and then it's asked to fill in this red square here based on the surrounding context. And that's sort of the standard mathematical modeling, or sorry, mass language modeling objective. Next. And so essentially, the model is basically trying to predict, which expression is, which expression should go in a specific, ordering based on the graph. Again, the standard mass language modeling objective. Next slide. All right, now I'll pass it off here to Rowan to go over some of the benchmarking results.
Hi. So I'll be discussing how we evaluate this model and how we know that it's a good model relative to the field of existing RNA foundation models. So as a reminder, this is how the field stands at the moment. There are three predominant models right now. There's a GeneFormer, SCGPT, and SC Foundation, and they all use transformers, although they're all slightly different and use different tokenization schemes. But what I'd like you to notice about this table is to take note of the parameter size column. GeneFormer has 30 million learnable weights, SCGPT has 50 million, and SC Foundation has 100 million, while our model has only 10 million. And just I keep that in mind. So we're a much smaller a model and potentially less expressive, or so you might think. But we're excited to show you how we evaluate it. The next slide. So we select three tasks for our initial evaluation. So cell type annotation, which demonstrates a holistic understanding of cellular states. Masked graph modeling that will demonstrate an awareness of underlying gene regulatory structure. And finally, a reverse perturbation inference that demonstrates understanding of genetic effects on the transcriptome. And this will actually carry potential for gene therapies and clinical application in the future. And our hypothesis is as follows, and this kind of ties together all three. So because we explicitly integrate a regulatory information into our model at the deepest possible layer, we hypothesize that we will have superior awareness of regulatory logic, and that will enable us to disentangle the perturbation responses that are mechanistically determined by the regulatory logic itself. So I'm going to go through all these in a little more detail and show you how we stack up. Next slide. So for cell type annotation, it's quite simple. The goal is to classify the cell type using the embedding of a foundation model, and we do this with a linear classifier on top of the output that's produced by a foundation model. And we do this on a data set of human immune cells and also a data set of cells that we did not see during pre-training. And I want to point out that for other models, we include a graph attention transformer module. And the reason that we do this graph conditioning is we will give other RNA foundation models the opportunity to see all the context that we've seen, so that if, say, our model is annotating cells better, we know that it's not simply a result of a wider conditioning context. Okay, and now we'll talk about masked graph modeling. For masked edge prediction, the problem statement is as follows. Given a regulatory graph, we want to ensure that the model is able to distinguish true edges from false edges. So to do this, we hide 15% of the edges in a graph at random. We feed it to the model, and then we sample gene embeddings, and we pass it through a linear layer, and we ask the model to distinguish edges that are actually in the graph. The blue one here, for example, the blue dotted line represents a true edge that's just been hidden, and the red line represents a spurious edge. And we just ensure we train the model to distinguish between the two. And we do this for human immune cells. We also do this for cancer infiltrating myeloid cells to assess its out of distribution predictiveness. And then finally, we have a reverse perturbation inference, and that is after a cell has been perturbed, and we are able to see its transcriptome. We then ask our model, or we train our model to reverse classify which perturbation the cell was likely to have received in order to induce the current cell's transcripts that we observe. And we do this with attention pooling and a linear classifier, and the attention pooling enables the model to focus on the most perturbation responsive genes that drive transcriptional response. And the data set we used for this analysis is the Adamson Perturb-seq data set, consisting of gene knockouts from the leukemia A562 cell line. All right. So to start with a graph structure understanding, we trained all the models. So we trained a taskhead on top of all the foundation models that I've mentioned. And on the left here, you can see that GREmLN has the highest area under the curve, indicating that it's the best at recovering masked edges compared to other foundation models in the field. And this is for human immune cells. And next we have myeloid cells. And again, GREmLN is in blue, and we see on the left that once again, it's the best at recovering masked edges. And as a reminder, all of these other models have seen the same graphs that we've seen, thanks to the graph attention transformer. Thanks to the graph attention transformer module that we include. And then finally, we go on to cell type annotation. Or first, here's just a quick visualization of GREmLN embeddings for human immune cells. And as you can see, it separates them according to the principal components plot. And on the right, we have the confusion matrix. And on the next slide, I'll talk about the benchmarking. And as you can see, for human immune cells and for these held out non-immune cells that constitute a zero-shot learning context, you'll notice that GREmLN is superior at annotating these cells, indicating it has the ability to distinguish cell types both in distribution and out of distribution. And what we'd also like to underscore is its parameter efficiency. That GREmLN is achieving better annotation despite an order of magnitude smaller parameter budget, 100 million versus 10 million versus 100 million from SC Foundation, for example, reflecting the efficiency of network guidance over scaling up and increasing model size. And finally, we emphasize again that we argue this evaluation is fair because we allowed other baseline models to see the same networks that GREmLN uses for diffusion via that graph attention transformer. And this underscores that GREmLN has a true mechanistic understanding of regulatory logic over regular transformers. And then finally, we have reverse perturbation inference. Oh, sorry, we have a quick ablation experiment. So in case you were wondering, how do we know that our performance is not on account of how we do tokenization differently, or a slight like difference in our wider architecture? We did an ablation experiment in which we used our exact same architecture, but omitted the query diffusion step over the graph. And on these 4 evaluations here, with GREmLN on the left and the version of GREmLN with the diffusion step ablated on the right, you can see that GREmLN is consistently showing a performance gain over its ablated counterpart. And this demonstrates that regulatory networks actually provide a complementary and mechanistic guidance that's beyond the hypothesis space of a transformer trained in isolation. And this really underscores the main contribution of our project here. And also, you might notice an interesting performance gain disparity, where for some of these evaluations the gap is huge, and for others it isn't. And what's going on here is that the graphs confer the smallest benefit in settings of high regulatory topology overlap between training and evaluation distributions. In other words, if it hasn't seen graphs similar to the ones that it saw during training, then the graphs provide the most benefit out of distribution when making out of distribution predictions. All right, and we'll move on to reverse perturbation inference. So as context, this is a visual.
Hi, let me make one comment here. This is Andrea Califano. Sorry for joining a little late. I've always been a little struck by the fact that some of the benchmarks that have been used to compare different AIs are actually essentially solved problems. You know, when you have an area under the curve of 0.957 and you're competing on getting, I don't know, statistical significance because you have a million cells, really the gain advantages are not obvious. So what we normally use are problems that are much, much harder to solve and where you can see a much more effective difference in the performance of different algorithms. And I think that's what Rowan is going to talk about less. In particular, I think the value of the next test is that it has true biologically relevant significance because it allows you to figure out what is the gene that is potentially mechanistically controlling a particular cell state. In this case, a cell state that is generated by a CRISPR perturbation, rather than just simply trying to classify cells, which, you know, sometimes when you want to know whether it's a CD4 or CD8 cells, you just look at the receptor or at the surface proteins, and we have great antibodies to do that, so we don't need an AI. But for the problems that Rowan is going to talk next, that is a really hard problem and one where I think the differences is more relevant.
Thank you, Andrea, for that comment. So yes, as Andrea mentioned, this is the relevant stuff that we're very excited about. So just as background, there's a perturb-seq data set produced by Adamson, and it's visualized on the left here, the UMAP, and it's colored by the different perturbation conditions. And what you may notice immediately is that they're not well separated. And sorry, also the problem of reverse perturbation inference, just to emphasize it, is to infer which genetic perturbation produced a given transcriptomic shift. And what you'll notice about the left is that CRISPR perturbations suffer from something called sparse transcriptomic shift. And that means that genetic perturbations often induce subtle transcriptomic changes that result in a low separability across conditions, which makes the problem of disentangling them a difficult one. So let's continue. So we did this for all the models, and here is our table of results. So we compared both GRN versions and PPI version of our model to other foundation models from frozen embeddings, and we found that the PPI model has the best out-of-the-box utility, and it's able to distinguish these perturbations with 30% accuracy. And when you fine-tune our GRN model, we achieve an accuracy of about 42%. And so a couple things to highlight. The protein interaction benefit is that the PPI version's superior frozen performance reflects that the first order effects of genetic perturbations arise from disruptions to the protein signaling pathways and binding partners, which makes intuitive sense. But this table also has something to say about the context-specific graph benefit, and that the GRN version has superior fine-tuned performance, and it highlights that context-sensitive graphs are essential to capturing the downstream long-range consequences of perturbation. And these results can also be analyzed through the lens of the bias variance trade-off. The PPI network is high bias, low variance, and it encodes universal signaling interactions that provide robust predictions out of the box, while GRNs on the other hand are low bias, high variance, however, and they require fine-tuning to fully exploit and learn from the regulatory heterogeneity that they offer. So let's continue.
Just
finally,
one more thing, which is also important, is that you can see that the other AI models don't really do any better than last logistic regression. So this is a hard problem.
Yes, that's right. And finally, just as a matter of interest, we also conducted an experiment in which we tried to recover the original gene expression on unseen data, and we found that we were able to do that successfully. And let's just to wrap up everything that we talked about in this talk, I'd like you to have these three takeaways about our model and what makes it special. The first is regulatory network integration, that our model presents a novel pre-training architecture for single-cell RNA-seq that integrates regulatory networks directly into transformers' attention mechanism. And the second is a matter of parameter efficiency, that incorporating said biological inductive bias, we achieve a deeper understanding of cellular states than existing RNA foundation models using only a fraction of their parameters. And three is mechanistic reasoning capability, thanks to the integration of the networks as well. And the network integration equips our model with a mechanistic understanding of the regulatory logic that enables it to successfully link genetic perturbations to very subtle transcriptomic shifts. And we're very excited about this. And finally, as future work, we have a few things in our pipeline, and that is efforts for enhanced explainability, such as interpreting the attention weights and the most active neural pathways that elucidate GREmLN's decision-making and reveal the biological logic that's driving predictions, and to dive into that. And the second is intervention design, in which we would leverage our model to assist in the design of genetic and drug interventions to induce desired cell states, particularly in an immune cell context, for example, reprogramming M2 to M1 macrophages in cancer. And finally, we have a project we have just begun that would integrate a language model interface using a model that was developed by the CZI AI team, RBio, and it would provide a language model interface for querying cellular states, and it would also allow us to connect with other knowledge sources. So that is our project, and we thank you for your attention, and we also would like to thank our corresponding authors, Andrea Califano, our PI and the BioHub NY president, Theo, the head of science at CZI, Mohammed Al-Qarashi, a professor at Columbia, Evan Paul, the AI platform lead at CZ BioHub New York, and Charlie O's, a researcher at Stanford, who is also an author on the paper. And finally, we're excited to answer any questions you may have. Yeah, and also if you want to access our preprint, you can do so here, and there's also a GREmLN a tutorial that you can use as well.